Hello and welcome to another Bioanalysis Zone coffee chat on the past, present and future of quantitative drug discovery practices. I'm Ellen Williams, digital editor of Bioanalysis Zone, and today I'm joined by Jose Castro Perez, Vice President of Product Management at SciEx, Dawn Duffield, Scientific Officer for Mass Spectrometry at KCAS Bio, and Vikram Shenoy, Senior Scientist for Quantitative, Translational and ADME Sciences at AbbVie. So thank you very much everyone for joining us today. So let's get straight into the discussion. So our ability to quantify substances at an incredibly small level has played a huge role in recent bioanalytical advancements. So firstly, do you want to expand on how we have achieved these improvements in sensitivity so far and how this has impacted the field of bioanalysis?
I can answer this question. So essentially, I would like to divide this into three buckets, right? The first one is sample handling, sample preparation techniques. We've seen a lot of improvements over the last, I would say, decade in terms of sample prep techniques around whether it's SPE, liquid extraction, whether we're using magnetic beads to extract whether they're small molecules, large molecules, immunoaffinity as well. They've all played a big role in sensitivity gains that we have observed, over the years. The other one is chromatography. Let's not forget that chromatography plays a pretty big role in LCMS. We've seen the shift from 4.6 millimeter columns internal diameter to 2.1 millimeter AD columns to 1 millimeter AD columns, and now even moving towards microflow rates as well, right? All of them show an improvement in signal as you move from the larger internal diameter columns to smaller diameter columns, injecting similar volumes. And finally, a lot of the work that it's been put into the development of new mass spectrometers, all the way from developing, what I call a brighter source. So, our ability to extract more ions, more signal out of the, X-ray droplet from improvements that have been made in terms of ion transmission from the source into Q1 region of the mass spectrometer. And also the last part, which I think is critical, is on the detector side, right? There's been a lot of improvements on the detector to, one, reduce the amount of noise, whether it's chemical noise, electrical noise, that will have an impact on signal to noise, but also our ability to detect more ions as well with the detector on the system. So I will say that it is a combination of all the three items that once you combine them, you can see enhancements in sensitivity that were not possible before. And I do see in the future that more improvements can be made as well in terms of other technologies, not just nominal mass per se, which is quote unquote triple quotable instrumentation, but also on high resolution accurate mass quant, because there are inherent properties of that analyzer that allows us to have better signal to noise and more selectivity specificity for certain analyze. So I think that what we're going to see in the future is more of a development in areas whereby we can look at enhancements that we can have from different types of mass analyzers that will have a positive impact on bioanalysis in general.
Lovely, thank you, Jose. So what techniques and technologies do we have at our disposal right now to tackle questions compared to maybe 10 years ago?
I can start with that one. I think I agree with a lot of what Jose just said. I think a couple things, that we're doing more routinely today is we have a lot more automated processes in place, which can help both on the upfront sample prep as well as on maybe automating some of the injections, even going to 96 or 384 well plate type experiments. And then I think really what's driving a lot of the work that I've seen done and in my history really is a lot of the sample prep using immunoaffinity or affinity capture. It's called many different things, hybrid, and those are becoming more routine. More people are using them, more people are getting familiar with that, more companies are using it, more CROs are using it. What that allows you to do is really clean up the samples. I think one of the interesting things that I'd like to talk about today is, how much further can we push the sensitivity of these instruments, right? We've been pushing, but I feel like eventually it's slowing down on the gains that we get with every next generation. And so how do we tap into leveraging, getting more out of what we have? And so for me, I'd still, part of today, maybe we can hear from the SciEx folks, some of that instrumentation piece, but the other part is really clean up, it's all about signal to noise, right? So if you can reduce your noise, reduce your background, you get more signal with less background, then you have more sensitivity. So I think that will be really critical. And then the other thing Jose touched on is high res mass spec. And I agree. I think a lot of companies, large, small, commercial, CRO, et cetera, are looking at these platforms for more targeted quant. I would say some of the barriers I see that still hasn't maybe taken off as quickly as we would like is the cost of those instruments are usually more, in some cases considerably more than an equivalent high-end triple quad or Q-trap. So if we can somehow get the cost down, and then the file sizes and the data storage kind of perspective is another kind of issue. And I guess the last piece that I actually think is an interesting, almost reversal, right? So I would say 10, 20 years ago, we were doing a lot of nano LCMS. And I would say in the last 6, 7 years, we've moved more towards micro, a little bit less nano. And I think there's different reasons for that. Sometimes the nano isn't giving us quite what we need, so the micro is sufficient enough. The instrumentation that's able to do nano chromatography becomes sometimes challenging. The ion sources that need to do it aren't as great, and maybe we're getting better sampling techniques, so we don't need to do nano because we're ionizing better and we're sampling more of what's coming out. So those are all things that I think, are where the field is kind of evolving right now.
I have a follow-up, maybe just a question for for everybody. My question is, is sensitivity enough? I mean, if you look at the next 10 years, we're seeing, very potent compounds like, cyclic peptides. They're extremely potent. They are hard to fragment. Very low circulating levels. Do we need more performance? Do we need more sensitivity? For instance, do we need better dynamic range? What, or is it enough? What we have today, for instance?
I would say you never can have enough sensitivity in my opinion, right? You can only see what you can see, and you're always pushing that. And this will go into maybe later part of our conversation, right? About ruggedness, because even if you have a great instrument that's super sensitive, as you run that instrument over the course of a batch or a day or a week or a month, the performance of that instrument typically goes down over time. And so then you're spending, and that can be fivefold, right? It can be significant. It's not, 20%. It can be significant. Now, usually that's not in a day, fivefold, but if it's a really dirty sample that you didn't clean up, it can be. And so for me, it's a combination of sensitivity with performance and consistency, right? Because if you're always fighting to get the best out of what the instrument can do, you're spending a whole lot of time spinning your wheels and working on things that hopefully could be easier. So I'm hoping, for me, that's an area in the future where you have less time where you're doing that and more time working on the science and working on, getting the methods where you want and driving what you need. And then maybe some of the sample preps help. But I definitely think we spend quite a lot of time on sensitivity or pushing it as best we can. And that includes large injection volumes, micro, nano, IPs, multi-chromatography, lots of different ways to push that. So anything we can help across any of those areas will make things better in the future.
Thank you, Dawn. And sorry, just one more comment or question. One of the real-time things that I see, at least from our experience, is developing a good chromatographic method as well as sample prep, right? I mean, on the mass spec side of things is a lot easier, I think. So do we see a future where, there is no chromatography? We just do sample infusion and we just take that out altogether?
Yeah, so I think today I don't see that working tremendously well because you have interferences. Now, again, you couple this with high res and you don't have, something that's nominally interfering, won't be in an exact mass interfering. Maybe there's a better potential there, whether you have no chromatography or you have 3-minute chromatography. But right now, we shoot for, let's say, in a routine, quantitative targeted kind of method, 5, 6, 7 minute methods. And we end up having to extend them a lot of times because of an interferent, right? So you have to get that chromatographically separated so that you can integrate the right thing. Or carryover. We're seeing a lot of carryover sticky things, right? So then you're spending extra washings. And again, one of the things that I want to talk about later as well is just the ability to, and again, this, I'll direct towards SciEx because I'm talking to SciEx, right, is all the vendors like to support their own platforms and they want you to, get their LCs and their stuff. And it makes it harder and harder to get the best LC coupled with the best mass spec so that you have the best answer. And I feel like if we move to a direction like Skyline, for example, where there's one platform that all the vendors are contributing to, and we can maximize that to the best of everybody's ability. And even if there's, service fees or something where you could still, from a company, be profitable and how you need to support that, but allow, users or your clients to leverage every tool they have at their disposal. Because right now we're limited. Like if I want to run a system that SciEx doesn't control, then I have to do contact closure, or I have to do something else, becomes very painful, and that doesn't help the situation. So I think, kind of went a couple different places there, but I think there's a lot of pieces there that, going to support things that we can eliminate chromatography when it makes sense, use it with high res, or simplify things or use better LCs or micro LCs, whatever it happens to be. I think it's going to get us, in my opinion, to the next level. It's going to really take that collaborative effort across many vendors to really offer something, unless somebody's got some magical solution in their back pocket on, giving us a 10x increase somewhere that I haven't heard about, which, I'll be waiting for ASMS. We'll see. Maybe it'll show up. But
in terms of sensitivity, I agree that with Dawn that it's never going to be enough. But yeah, I do agree that a big part of that is focusing on reducing noise, not just boosting the signal. And then as drug compounds are just going to get more and more potent, right? So I think just keeping up with that, as we go lower and lower doses, we're just going to need to keep up with that on the hardware side and software side and sample prep as well. So yeah, I think sensitivity will continue to be a growing need. And on the throughput side as well, I agree. I think I don't see right now that we'd reach a point where we're not going to need chromatography, but it really depends on what innovations we see on front end technology, as well as anything we're kind of taking away from the instrument side. So if we're reducing time on the instrument side, typically we need to increase time on sample prep, right? So it kind of just shifts the burden elsewhere in the workflow. So I think as long as some of those capabilities are improved and some of those lingering questions are addressed, that would enable a future in which we're not doing chromatography and really improve the throughput.
Great, thank you. It was lovely to hear each of you comment on that. Thank you very much. So we're in an era where personalised medicine and patient-centric sampling are all coming to fruition. But what do you think are some of the breakthrough highlights of the last decade that have maybe made these possible?
Yeah, so maybe I can start with that. So I really think this personalized medicine is kind of started with the advancements in just science in general, genomic data, understanding individuals' personal mutations, and then, maybe gene therapies, which kind of then have targeted approaches. I think, again, from the mass spec side, the fields that those open up is, doing some of the illegal work. And then you start getting into ion pairing. We were just talking about chromatography, right? Getting into ion pairing agents and how do we get around that? And is there ways to improve sensitivity there? I think a lot of, we were talking earlier about needing, super potent compounds and more sensitivity, but we also have a lot of what I would call conjugates, whether it's an antibody drug conjugate or an antibody siRNA conjugate, or just a complicated fusion protein with something else. And what that does is allows maybe targeted delivery to a specific, tumor type or disease state. You have extended releases, which then, make you, your drug lasts longer, but then eventually goes down, and you have to have the sensitivity to see those. They become much more complicated bioanalytical problems. And so you need to then maybe look at multiplexing different parts of a molecule and getting a lot of data in one go. So again, having instruments that allow us to do that. Then you get into, microsampling, and patients able to sample themselves at home, just prick, like almost like a glucose test. Like you prick your finger, you prick your, arm or whatever, you collect 20 microliters of plasma or blood, and then you ship it away, right? So it's quicker, it's more convenient for home use, but that brings up a whole area of technology that now we have to develop bioanalytical methods that will allow us to extract, almost like the dried blood spots, but now it's more of a still maybe liquid phase, extract similar analytical analytes, I guess, out of smaller sample sizes. So one of the ways you get increased sensitivity is you use larger samples, right? So if in theory you had a sample that was 20 microliters and you got 10 nanograms per mil sensitivity, if you needed 1 nanogram per mil sensitivity, maybe you take 200 microliters or something, right? You can scale that up, and with some of the sample prep approaches, concentrate and do different things, whether that's through IPs or dry downs, etc. Now with microsampling, you can't do that anymore, right? Now you're only limited to 20 microliters total. So then that goes back to really needing and pushing the sensitivity of the instrument. So I almost think it's going to come full circle, right? In one case, we're going to have smaller samples, which makes this more flexible. We can have personalized medicine. We can, get samples at home, but now we're going to need better instruments and techniques to allow us to get to the same point we were before, or even more if they're more potent compounds, or more needs for that kind of sensitivity.
Great, thank you, Dawn. So the primary focus of today's session is to discuss where the field of drug discovery and bioanalytical testing is heading in the next 5 to 10 years. You kind of touched a little bit on that in the last question, Dawn, but are there any specific technologies or technological advancements that you anticipate will lead in the field of bioanalysis in the coming years? Let's maybe go to Vikram first.
Yeah, well, one thing that's kind of been trending in my mind is I think the capabilities of mass spec imaging are something that's really caught my attention. It's all the buzz, right now. And I think it really speaks to the versatility of mass spec platforms. And in kind of, I guess, taking the conversation beyond sensitivity and throughput, I think, spatial information is something that's lost in a lot of the kind of bread and butter workflows that are employed in drug discovery. So I think, seeing kind of the complementary information and data provided in these mass spec imaging, workflows is really exciting. And I think that's going to be one major thing to keep an eye out for in the next decade.
So maybe I can ask a question there, Vikram. So we've looked a little bit at imaging as well, but I guess in general, are you thinking more from a discovery, understanding maybe the mechanism, the pathway, as opposed to, a high throughput quantitation type approach? Because obviously I work at a CRO where we're looking at throughput, right? We're looking at instrument cost, and where I see imaging is more in those less global applications, right? A little bit more niche, but can again provide a lot of power and a lot of information. But it's hard for, let's say, the general public, even the people that know a lot about LCMS, to jump into that field, because it is such a specialty area. So what are your thoughts there?
Yeah, I mean, I think it's more in the applications of understanding disease mechanisms, understanding, but also, with looking at at drugs specifically, their distribution within the tissue, with definitely with small molecules, that's a bit more established. I think, with me coming from the large molecule side, I think there's a lot of development going on there to enable imaging the distribution of large molecules within tissues. So I think it's early enough that we'll see more and more applications be established in the near future. But yeah, it definitely.
Go ahead.
Sorry, it definitely is. It's not something that's suited to kind of the high throughput, world of bioanalysis that CROs see right now.
Yeah.
And it definitely is for more niche applications. But yeah, I think the fact that it's complementary to that is really powerful.
Yeah, and I think one of the nice things that always gets asked when you're doing, let's say, tissue distribution studies is it a homogeneous distribution, right? So if you take different parts of the tissue, is it all concentrated in one part, let's say, of a brain? Is it specific to the cerebellum, or the cortex, or some other part, or is it all over the place? And even within a place, is it heterogeneous? Is it homogeneous? Because at the end of the day, when we sample in a more quantitative way, we take a section, right? We take a piece, or sometimes we homogenize a bigger section, just kind of depends. But are we getting a representative sample? So I do think imaging probably could help answer those questions a little bit more.
I think
Yeah,
that I think that just going back to this question, I can think of a couple of areas here. And certainly one of them is around, biomarker discovery, right? I think that, again, if you look at all the work that's been done in the past around, deep visual proteomics or deep visual metabolomics, having that actual deep knowledge and understanding of biomarkers, right, at the tissue level or even at the single cell level, how are we going to be able to interpret that data? I mean, there's a lot of data points, and especially if you're looking at that single cell level, I think that's where there are certainly gaps in the workflows and technologies to be able to not just visualize that data, but also understand the data. So I think that we're going to touch upon that later on in this coffee chat. But I think that this is where artificial intelligence plays a big role to allow us to really understand, these data streams, these data lakes, right, of information that we need to generate to, understand the biology, understand the biomarkers, understand the disease in more in depth, right? Not just from the PK standpoint, but also understand what else is happening.
Lovely, thank you. What current practices then might need to evolve to align with some of the field's goals for the next decade?
So again, I can maybe start with this one. I touched on this earlier, right? I feel like we really need to get more rugged, consistent instruments that the performance doesn't change quickly over time or even at all would be ideal, right? That you have an instrument and it just almost becomes like a plate reader, right? You get it, you get a signal, you run, you don't have to spend all this time in method development and optimization. And then the other big piece again for me is that flexibility between vendors, right? Because the more and more complicated modalities we work with, the more challenging the bioanalytical is, the more we need to put our best instrumentation across, kind of irrespective of vendor platform. And right now I find, certain vendors work with other vendors nicely and other ones are more their competition and they just don't support those platforms as well. And so having the ability to have maybe a more universal platform, again, I don't know if it's ever going to happen, but I think that's where it really does need to evolve. We need to have an evolution of the field. And I use the example of Skyline, right? In the past, there was a lot of software. Each vendor had their own software and they still do. But now we have this Skyline software that can be used for quite a bit of work. And I think most of the vendors have contributed to the development of that. It obviously started in a university setting, but now it's being supported by a lot of others. And I think things like that allow you to, again, get all the best and brightest minds and people getting us where we need to go. Because if we really want to advance science properly, there needs to be a healthy competition, but there also needs to be a nice collaboration. And I think getting the right balance between that is maybe the biggest challenge.
Are there any emerging global trends that you think may impact the industry going forward?
Yeah, the one that I'm thinking about now is the 3 Rs, which involves the reduction of animals in testing. So coming from the preclinical side, that's something that's always top of mind. Is how do we get more data with less animal usage? And so that's going to kind of take, what we might expect then is lower sample volumes. And so what does that mean? That means we need more sensitive instruments. We need to be able to work and get more data out of less sample volume. We'd need better liquid handling tools to pipette, microliter or single microliter or sub microliter sample volumes. And just like more robust instrument systems, because we only have one shot to get all the data we need from this little amount of sample. If something kind of interrupts the run, and then we lost the data from a really precious sample, that'd be tragic. So that's definitely a trend that I think will push our field to its limits.
Great, thank you, Vikram. Maybe Dawn, let's go to you for this next one. So from a technology perspective, what potential barriers do you see that could hinder bioanalytical progress in the coming years, and how can we preempt these?
Yeah, so again, I maybe start to sound like a broken record, but I feel like a lot of this is things we've talked about, right? Good hardware, good software, reproducible, rugged instruments, flexibility across platforms, flexibility. A lot of times, these instruments are very expensive, even if you're buying 1 or 2 or whatever. And if you're at a shop where you're in production and you're buying 10 or 20, it's even more. So having the ability to have them set up and easily transition from application A to application B 2 days apart or 3 days apart, and not dedicate them to nanospray, for example. Like in the olden days, let's call it, we used to do a lot of nanospray, but you don't want to go back and forth, right, between nano and high flow and nano and high flow, because it does become a little bit of an art form. So if we can get away from it being so much artful and a little bit more rugged and, you know, production mode reproducible, or flexibility. Like if you're running oligos one day where you have to use ion pairing agents to help your chromatography, you know, those take a while to get the system running. They can coat the rails, you know, they become more problematic for cleaning. And so you can't just in the morning do one and in the afternoon do something else. It might take 2 days to clean that out. So if there was ways to, you know, streamline jumping from different platforms, or again, you know, you can do oligos with, let's say, a Hillock chromatography that doesn't require ion pairs, but in general, when you do Hillock, you lose sensitivity. So if we had something that had 10-fold more sensitivity, maybe we could just do Hillock and say, okay, we don't need to push, you know, the ion pairs to keep the sensitivity. So it's, for me, it's that whole culmination of everything that's making things a little bit more streamlined, more flexible, more easily translatable across platforms, across vendors, across modalities, you know, increased mass ranges, let's say for triples, you know, where you could go higher and not have to go to high res. Or again, you know, cheaper high res, so that you could do that more routinely and not have to always pick, you know, only one or the other. So those are some of the things that, again, I always, myself personally, will push vendors to work and come up with creative ideas as best they can to help all of the rest of us, you know, be able to answer the questions we have to answer on a daily basis to, you know, advance science and medicine.
Lovely, thank you, Dawn. So Jose, you did touch on this a little bit earlier, so maybe let's go to you first. But with automation, artificial intelligence, and data analytics becoming very significant aspects of bioanalytical testing, where do you see these tools being applied in future, and how might they need to be optimized to keep up with further advancements?
Yes, I mean, we're now seeing, you know, automation, artificial intelligence everywhere. Um, Even the other day, I had a Microsoft 365 Copilot, and I used it for the first time. I was actually quite impressed with it. Um, you know, this is something that we're going to see more and more of, and I think that not just from bioanalytical testing, but also from a drug discovery perspective as well. So I personally think that, you know, one of the biggest bottlenecks that we have right now is twofold, right? The first one is, you know, compound optimization. You know, if you're doing drug screening and you need to optimize compounds for, you know, mRNA transitions, for example, you know, how do you know what is the best transition that you need to pick? How do you know, what are the best conditions in terms to get you the CVs that you want to get, the dynamic range of the assay? Wouldn't it be great if you just tell the software, hey, here's my list of compounds, these are all my structures, these are my conditions, go and figure it out. And you spend more time solving other more important problems, right? And that's from a, you know, component optimization standpoint. From a data processing standpoint, you know, we do spend a lot of time just looking at integration, right? Just, oh, we've got a shoulder here, you know, you need to go and modify this peak here. And, you know, I was actually quite impressed, you know, when I visited a number of labs in Europe, and they were quantifying like 300, 400 pesticides per sample. You can imagine having 400 MRMs. And if you need to look at one by one, and you have a plate of 96 samples, it's a daunting task. So to me, that's a routine bioanalysis that we underestimate how much time we actually spend looking at the data rather than spending more time trying to solve other problems, whether that can be automated as well and really reduce the time to answer. Um, you know, I think that there's a lot of opportunities here to improve the overall, I would say, you know, data integration, optimization, is that overall feel, you know, connectivity with the instrument between the bioanalyst and the instrument and, you know, the data output. I think there are a lot of opportunities to improve the overall workflow. And if, technology advances in a way that, we were talking about high res, accurate mass quant, for example, whereby you don't need to do any optimization, right? Everything's done for you, single injection, single scan mode, and you can have all the data there all the time. So you can go back a year later and the data is still there, all the data, not just the amortization that you're looking for. So with the right software tools and the right workflows and ecosystem, I think that can become a reality. And I think from a productivity standpoint, we can spend more time, addressing problems or even developing new methodologies as well that we need to work on in the future, or just doing research that we couldn't do, right? Because we just already burdened just doing, optimization and data processing. That's where I think that, there's a real need to advance our interaction with LCMS.
Yeah, I would echo a lot of that. I agree. We spend a lot of time integrating, large batches with many MRMs, or, high res is even more complicated, right? Because then you got lots of more data to look at. If there's ways, even today, right, there's ways to say, have the software tell us what's the best integration algorithm. Try 2 or 3 in the background and tell us across 95% of the samples, this one fits better. Even today, we have to do one and we can't even easily change it. We have to like make a whole new method and then start all over to try the other one, right? So it's not like you can go from auto peak to multi-quant 4, or some type of differences. And so we spend a lot of time redoing that data. So if AI or some type of more automated data processing could help with that, it would save a lot of time. And then, even picking the best curve, like is quadratic 1 over x the best, or 1 over x squared, or should it be linear, or are there other choices, having it automatically show you that based on data, almost like real-time review, so you don't have to do it, would simplify things quite a bit. And then, that to me seems pretty easy, like easier to accomplish than some of the other stuff, which might be coming up with automated programs on the method development, right? How do you get a program to tell you, what's happening with your system when something's not working, like a troubleshooter, is there some way to help you troubleshoot so you know what part's not working? Is it your mass spec? Is it your LC? Is it your this? How could you have it suggest different gradients to try to tease out carryover, when you have something co-eluding and you want to keep your, you put in parameters. I want the shortest runtime. I want the most sensitive with baseline resolution in, this window, something like that. And you could say, tell me what to do to get that, or mobile phase, implications of changing mobile phases, all of that stuff, I think feels a little bit maybe harder because it's experimental and software. But something like that, I think would really change, I think, the future of how we're doing some of this stuff today.
Lovely, thank you. And to wrap up then, what developments or changes are you most excited to witness over the next 10 years?
That's a hard question. I think everything, right? Making it, all of the stuff we talked about, making it less of an art and more consistent, more predictive, more quicker, spending less time, being more efficient, being more sensitive. I think all of that, and maybe seeing it all come together, I think will be very interesting over the next 10 years.
I agree, Don. I think that the other point that I think that we touched upon before was around personalized medicine, patient-centric approach, bringing the technology and the patient closer together, right? I mean, after all, we do what we do because we want to improve the quality of life of patients. So I think that the technology overall still go ways to go. We need to build more intelligence in the way that, not just instrumentation, but, the processes before we actually, put the sample in the mass spectrometer. I think that, we spoke about sample prep, we spoke about maybe no need to have separation, right? Removing moving parts. there's a lot here. I think it's quite exciting to see the pace at which technology is moving. And as you mentioned as well, the potency of compounds, they're getting more and more potent. There are more challenges. We're getting less sample as well. So there's a lot to be excited about here in the next 10 years.
Yeah, and one more thing just came to mind, and maybe this is applicable to an earlier question, but I think again with patient-centric sampling or small samples, we are required and asked more and more to do multiplexing. So more analyses in one sample, get as much as we can, all the information we can get. And sometimes, let's say you're doing a biomarker panel that has 10 different biomarkers. You have some that are screaming, saturating the system, and some that may be right at the edge of the LLOQ. And so if, again, from an AI perspective or an automated software platform, if there was a way to say, let's attenuate this signal, and keep this one as much as we can in a automated energy, change DPs, change collision energies, whatever, in a more automated way, so that all of those samples could be run together and not be saturating in one area and not in the other. Those could be an interesting, advantage that would help us move forward in those situations.
Yeah, I think a lot of what I would want to say has been said already. But yeah, to reiterate, I think every time I think we've reached a new height, and I'm just starting to get into the newest technologies that are out there. Something else happens, something else gets developed. Like there's a new way to new fragmentation methods. There's new front end separation techniques. There's better sample prep that's happening. There's increases in throughput, and we've kind of touched on all of that throughout this chat. But I think me being in San Francisco, I have to give. I'm a little biased towards AI and automation, but at this point, for me, that's because it just feels like a magic wand, and it just sounds like it's going to really revolutionize the field in terms of predicting more and more complex unknowns with better accuracy, and really taking the burden off of the wet lab. It'll be able to simulate experiments, and of course the conversational aspect of just having like a co-pilot or a chat bot, integrated into every piece of software that we use, so that we just use natural language to say, this is what I want, without getting into needing to know all the different terms or to speak a very specific way. Just having some software like that can understand you really unlocks the capabilities. And I know it's because vendors put so much time and effort into adding so many different capabilities. It's impossible for us to all be knowledgeable about all of it. And I think that's where personally AI will make such a huge difference in the near future, let alone what it can do beyond the next decade.
I mean, I think I'll be the maybe devil's advocate kind of cynic, right? You always want to balance getting the most out of something, but also, there's real people in the world that are fearful of them then taking over and not being needed, right? So it's always like you want it to do as much as you can, but you don't want it to get so much that maybe you become obsolete yourself, right? So I want to stick around for a little while. So hopefully we'll work together and make a good companion.
Well, I think that's a great place to finish up. So thank you very much to Jose, Dawn, and Vikram for providing your insights on this topic. It's been a real pleasure to speak to you all.
Thank you.
Likewise. Yeah.
Thank you.
Thanks for having me.